Okay, class, now we're ready to get into chemical digestion. Okay, let's look first at some of the carbohydrates. Okay, this right here is maltose. So we're looking at disaccharides. Maltose. Maltose is glucose covalently bonded to glucose. Table sugar, sucrose, is glucose covalently bonded to fructose. Okay. So we're showing what you have to break apart. And see, maltose would be like in breads, beers. Sucrose, is table sugar. Okay. Here's some disaccharides. Sucrose, maltose, lactose, and milk. Galactose bonded to glucose. All right. So what we need to do then is, let's go over here. Digestive enzymes, salivary amylase, pancreatic amylase, and brush border enzymes in the small intestine. Dextrinase, okay, glucoamylase, lactase, maltase, and sucrase. So that's what we're looking at. These enzymes here are supposed to break these down. Okay, let's look at it. Sucrase, these are brush border, so that's in the small intestine. Sucrase breaks down glucose to fructose. Okay, that's sucrase. It's brush border enzyme. Maltase breaks down maltose, and lactase, okay, breaks down lactose. See, some people have lactose intolerance because they don't have this enzyme sufficiently made. Therefore, this wouldn't get broken down. And remember, you need a monomer, so it would go into the large intestine. Remember all the bacteria we talked about? They love it. They started making gas and diarrhea and everything. Okay? So we'll go a little further. Okay? So here's amylose. Remember, this is plant. Now right here, this is how plants store their carb, one of the forms. So amylase here breaks down amylose. It breaks those 1,4 linkages, trying to get the monomer. Okay. Right here is amylopectin. That's the other form that plants store their carbs. All right. Now, what we need to do here is we would use amylase to break down the 1,4s, but this is a 1,6 linkage. So what do I need? I need a dextrinase. Okay. An alpha dextrinase. All right. Remember, enzymes have specificity. So we go a little further. Monosaccharides bound together by the removal of a water molecule. Okay. So this was just showing how they are formed. See, a removal of dehydration synthesis to form covalent bond. That bond is called a glycosidic bond. When two more come together, disaccharide. When 3 to 10, we call it an oligo, and greater than 10. That's good etiquette. Good etiquette. Okay. So we go further. Sucrose, table sugar. Glucose, we just did that. Just hitting it again. So here are beer, maltose, lactose, sucrose. See right there. Okay. That's amylose. Amylopectin. Okay. Alpha 1,4 and alpha 1,6. That's where branching. See, this molecule is branched. That amylopectin. Okay. Then, of course, we have cellulose. Cellulose you don't break down because it has a beta bond in the cellulose. It adds bulk to the diet. The plant wall, the cell wall. See, a plant has a cell membrane and a cell wall. The cell wall of a plant is like its skeleton. That's what makes it stand up. So when we eat the plant, we can get the juice out, get the amylopectin and the amylose, but not the cell wall. But the cell wall adds bulk to the diet. See, the smooth muscles in there, just like you go to the gym and lift weights, the smooth muscles need to have something too, because if everything gets digested and absorbed, what do they have to work against to keep strong? what do they have to push against as far as on the carb? So that's what that fiber, you know, you've heard of Metamucil and so forth. Okay. See, here's amylose and amylopectin, starch, a plant polysaccharide, whereas glycogen is ours. Okay. So, chemical digestion, absorption, digestive end, salivary amylase, pancreatic, brush border. The amylase breaks alpha-1,4 linkages. The dextrinase, as I mentioned, breaks 1,6. Now, glycogen has a lot of 1,6s. Glycogen has a whole bunch of 1,6s. Let's see if I have a glycogen here. Should have a glycogen. There's glycogen. Glycogen is heavily branched. So you need the 1,4s and the 1,6s. That's what the dextrinase. Lactase, we already said. Okay, see, all of these here are to try to get to the monomer form. You can, you know, go and study them and write them down. Okay, let's go further. Chemical digestion and absorption of carbohydrates. Okay. Let's go here. Okay, so we talked about that. Okay. Now, chemical secondary active transport, co-transport with sodium. Facilitative diffusion of some intercapillary beds in the villi. See, here's how this comes in. Absorption, secondary active transport with sodium. Remember, we talked about that sodium does secondary. You have the sodium-potassium pump, which keeps sodium low inside a cell. Sodium's going to leak in. When sodium leaks in, it's very exergonic because remember the inside of the cell is negative and sodium's positive. So that secondary transport means is secondary to the fact of having the sodium-potassium pump. So when sodium comes in, it can bring some of these nutrients with it. So absorption of carbohydrates, that means these monosaccharides, secondary active transport with sodium, facilitative transport of some monosaccharides. Okay. Enter the capillary beds in the villi. Okay. Remember, facilitative is a help diffusion. Glucose does like that. So that's how the absorption, and then it goes on into the capillary. Now here's an amino acid here. These are looking at amino acids. We haven't got to amino acids, but the carbohydrates, some of them also use the monosaccharides, and some use facilitative diffusion. Okay. So protein digestion and absorption. Dietary proteins, that's about what you have. So you're bringing some in the diet, some in the gut, and slough mucosal cells. We're seeing where we get these amino acids from. Now remember, proteins have peptide bonds, so we need to break those peptide bonds in order to get to the basic amino acids. So that's where the proteases come in. So let's go here. Okay, chemical pepsin in the stomach, pancreatic proteases, trypsin, chymotrypsin, carboxypeptidase. Then we have brush border, what's aminopeptidase, carboxy, and dipeptidase. Okay, absorption is coupled to sodium, as we mentioned. All right, let me kind of explain this. All right, let's see if we can find something here. All right. This is why we have so many different proteolytic enzymes. All right. We're going to break them up into what we call exo peptidases. Now let's make this protein big here. We're going to break it up into what we're going to call, here's a long protein, let's say, exo peptidases and endo. So this is why we have, you might think, redundancy in some of these. An exo peptidase breaks from the end. We have an amine end and a carboxyl end. So the aminopeptidase cuts one by one from this end. And the carboxypeptidase, okay, carboxypeptidase, from these are pancreas, brush border, let's say, cuts from this end. So that's the exopeptidase on the outside. The endopeptidases break peptide bonds within the protein. Pepsin from the chief cells breaks bonds between the amino acid tyrosine and phenylalanine. Now, am I going to get that specific? No, but I'm just showing that that's where it cuts. So if there's a tyrosine covalently bonded to phenylalanine, it'll cut there. Trypsin from the pancreas cleaves pep mainly at the carboxyl ends of between lysine and arginine, two more, except, okay, chymotrypsin cleaves bonds where the carboxyl side of an amide is a tyrosine, tryptophan, or phenylalanine, and dipeptidases split the dipeptides. Okay, so each of these is not redundancy with all these coming from the pancreas, the brush border, and the stomach, is what their specificity is. But ultimately, what you're trying to do is break them down into individual what? Amino acids. And then that shows when you break them down that this secondary transport helps bring these amino acids in. And then at the back here, you have amino acids leave the epithelium by facilitating diffusion and enter the capillary. So it's a help transport here. Okay. That's how you get those in. Okay. The chemical digestion and absorption of lipids. Okay. We're gonna start a new video for the lipids. So what we have done are proteins and carbs. We've done proteins and carbs.